Okay, so welcome to this next video in which we are discussing the ligand binding domain of the nicotinic acetylcholine receptor. So in the previous video, what I said was we were trying to show that cation pi interactions were important for acetylcholine binding to this aromatic nest and binding therefore in this ligand binding site of the acetylcholine receptor. Okay, now what we want to show is that that is the case. So how can we do it? Well, we can remove the delocalized electron rings from a tryptophan residue which is in that aromatic nest. And if we do this via adding progressively more fluorine atoms onto the aromatic ring of indole, okay, then the number of electrons in the delocalized ring goes down. They become less negatively charged, and therefore their ability to interact with the acetylcholine's cationic charge, it goes down. So what we should see is a drop in the affinity of the receptor with this modified tryptophan in as you go down and make the modification more severe. And that is what you see, but I want to now explain to you how it is that you can get a single modified tryptophan in that exact position. Okay, and this is something known as the in vivo nonsense suppression method. Okay, and you'll see why it's called the in vivo nonsense suppression method. And specifically, it's the in vivo nonsense suppression method for expressing unnatural amino acids. Okay, so in vivo nonsense suppression method for unnatural amino acid incorporation. But all that means is we are trying to get this modified unnatural amino acid to be incorporated at just one place in the entire protein. Okay, so how are we going to do this? Well, basically, what we do is we go to the gene for our protein. Okay, so we're trying to replace a specific tryptophan. So we know where this tryptophan is that we're trying to replace. So we know which codon or which triplet of organic bases in the gene corresponds to that tryptophan. Okay, right, so let's say this is the gene for our protein. Okay, so this is the gene. Right, and we know what's going to happen. I'll just remind you of the central dogma of biology, just to make sure everything's crystal clear, because suddenly we're going to have a very clever portion of this. So this is the gene. Okay, now the gene is transcribed to mRNA. Okay, so on this DNA, one of the strands is the coding strand. Okay, so let's say in this turquoise color here, this is the coding strand. Okay, and what we will do is we will create a piece of mRNA that is complementary to the coding strand. So here is our piece of mRNA, which is complementary to the coding strand of the DNA. Okay, so it will have the same sequence of organic bases as this non-coding strand of the DNA, except that obviously it will have uracil instead of thymine because it's RNA rather than DNA. Then what you will do is you will transcribe this translate this mRNA into the protein. Okay, so here's the ribosome, here's the mRNA, here's your protein coming out. So basically, the tryptophan at some point of this protein will have had a codon that corresponded to it. i.e. there will have been a triplet of organic bases on this mRNA. So let's make this bigger. So there will have been 3 organic bases, 1, 2, 3, which were read by the ribosome and were interpreted as meaning we should put a tryptophan in at this specific position. And this is the tryptophan we want to alter and put in our modified tryptophan instead. So what we do is we alter this codon. Okay, we turn it into a stop codon. So we are going to turn this into a stop codon. And you might think, what on earth? But there's an ingenious there's an ingenious point coming. So turn it into a stop codon. Okay? Right, and this is where this idea of nonsense suppression is coming from. Because a nonsense mutation is where you turn a codon that wasn't a stop codon into a stop codon that causes premature halting of the protein synthesis, and that's called a nonsense mutation, because you're not going to make any sensible protein. Okay, now what example do we have of a stop codon? Well, a stop codon example that we could have is UAG, uracil adenine guanine. Okay, so what we'd of course have to do is we'd have to do this on the level of the DNA. So in order to turn this codon into a stop codon, we'd have to alter the coding strands so that it had the correct complementary sequence of organic bases to this stop codon. So this green strand would have to have the sequence ATC, which is the complementary DNA sequence to this RNA sequence. So adenine is complementary to uracil, thymine will be turned into adenine, cytosine into guanine. Okay, so we will alter that triplet of organic bases which corresponds to this codon which is making our desired tryptophan, and we will alter it to ATC so that we then get a stop codon here and the protein is going to be prematurely stopped. Now, this doesn't look at all helpful, except that there exists a species in the world known as tetrahymens. Okay, so tetrahymens Thermophila. Okay, and this species does not use the UAG codon as a stop codon. So tetrahymens Thermophila basically does not use this sequence of organic bases on mRNA as a stop codon. So instead, it has a tRNA with the complementary anticodon to this, because usually in vertebrates, when you have a stop codon, you have no tRNAs with complementary anticodons to that stop codon, and that's how it triggers the halting of the translation. Because if you've got absolutely no tRNAs with the complementary anticodon, then you just cannot bring tRNAs in which will add amino acids on for that codon, because there aren't any which have the complementary anticodon. So what you do is you go to your tetrahymens Thermophila, and basically in that species UAG is not a stop codon. So tetrahymens Thermophila does have tRNAs with the complementary anticodon, which in this case is AUC, AUC. Okay, and basically what you can then do is you can take these tRNAs with this complementary anticodon from the tetrahymens Thermophila, you can stick on your modified unnatural amino acid. So here's our unnatural amino acid. So in this case it will be this modified tryptophan with the fluorines on. So this is our unnatural amino acid. And now what we can do is just put this unnatural amino acid with its tRNA in. And now what will happen? The ribosome will let this tRNA come in, and it will put in our unnatural amino acid in place, in place of this stop, in the place of the protein that this stop codon corresponds to. So when you reach this stop codon in the ribosome, either the ribosome will basically put in our unnatural amino acid, and then it will continue on with the protein synthesis, because everything else has been left untouched. All we did was alter this one triplet of organic bases to turn it into a stop codon on the mRNA level. Okay, so basically either it can make the protein with our unnatural amino acid in, which in this case is our modified tryptophan with its fluorines attached on, Or what else can happen? If it doesn't use the tRNA, our new tRNA from tetrahymens formophyta, which we've stuck our desired amino acid on the end, what will happen? It will just put a stop code on that and it will terminate it. So either it doesn't make any protein at all, or it makes the protein that we have told it to make, basically. So this is a way of ensuring that you only now produce nicotinic acetylcholine receptor subunits which have this modified tryptophan in. So that's how we can produce nicotinic acetylcholine receptor subunits which have this modified tryptophan in the acetylcholine binding site. And basically, as you make the tryptophan's modification more and more extreme, as you reduce the number of pi electrons in the pi orbitals, then the affinity of the acetylcholine to bind to the nicotinic acetylcholine receptor, acetylcholine binding site, becomes lower and lower and lower, and it's because these cation pi interactions are becoming weaker and weaker as you deplete the number of pi electrons in the pi orbitals. So that shows that these cation pi interactions are very important. And I think in some ways the in vivo nonsense suppression method for unnatural amino acid incorporation is in some ways more important than the actual understanding of the nicotinic acetylcholine receptor, because this is a really, really clever way of incorporating the exact amino acid you want. And the science may well change. Our understanding of the nicotinic acetylcholine receptor may change. We don't know. But the method is true science. So I hope you've enjoyed seeing the experimental method.